speaker is Qianru Hui from the University of Manitoba, and she will be speaking on the effects of L-tryptophan and 1,25-dihydroxyvitamin D3 on the osteogenic differentiation of mesenchymal stem cells, I don't think I said that right, isolated from compact bones of broilers and layers. So, Qianru, please go ahead.
Yes, can everyone see my screen?
Yeah, you're great. Go ahead.
Oh, sounds good. Okay. Yeah, thank you, Brenda, for your introduction. And thank you for attending. I'm Qianru Hui. I'm a PhD student and PhD candidate from the Department of Animal Science, University of Manitoba. It's my honor to be here to share my study. So today I'll be talking about the effects of L-tryptophan and 1,25-dihydroxy vitamin D on osteogenic differentiation of mesenchymal stem cells isolated from the compact bones of broilers and layers. Well, I will be going through 4 parts to introduce my study. Bone problems are a big challenge in both broiler and egg industries, causing huge economic loss and animal welfare issues. Skeletal disorders such as tibia dyschondroplasia, rickets, and egg deformities in broiler chicken are mainly attributed to rapid growth, and laying hens mostly experience osteoporosis or bone fracture during the egg laying period because egg production constantly withdraws calcium from medullary bone for eggshell formation. Bone mesenchymal stem cells are known as strong self-renewal ability and multilineage differentiation potential, which can differentiate into osteoblasts, adipocytes, chondrocytes, and myoblasts. They continue contributing different types of cells to bone formation and playing an important role in bone turnover and metabolism. A calcium sensing receptor has been reported to be involving osteogenic differentiation and is a potential therapeutic target in osteoporosis. Calcium sensing receptor belongs to class C GPCR, and it can sense subtle changes in extracellular calcium concentration and thus mediating parathyroid hormone secretion to maintain calcium homeostasis. In addition, CSR mediates a variety of physiological and pathophysiological processes. While the binding of CSR with its ligands leads to CSR conformational changes and induces biased agonism and antagonism via downstream G protein coupled signaling. Moreover, L-tryptophan has been recognized as a potent human CSR positive allosteric modulator. L-tryptophan as an essential and functional amino acid is involved in both protein synthesis and the neuromodulation process. 1,25-dihydroxy cholecalciferol, which is active form of vitamin D3, is important in calcium metabolism because many genes related to calcium transport and calcium metabolism have vitamin D response element sequence located in neuropromoter region, thereby acting as a transcription factor of these genes, including CSR and many calcium transporters. Well, so the hypothesis of this study were CSR nutritional modulator and 1,25-dihydroxy cholecalciferol promote osteogenic differentiation of chicken mesenchymal stem cells. Well, the objectives of this study was to compare the differentiation processes of mesenchymal stem cells isolated from both broiler and layer compact bones, and also to review the regulation mechanism of L-tryptophan via CSR during chicken mesenchymal stem cell osteogenic differentiation. Well, the first step for this study was to isolate mesenchymal stem cells from compact bones of femurs and tibia of 14-day-old Ross 308 broiler chickens and also 14-day-old decoy pullets, and then verified the isolated cells. Well, the mesenchymal stem cell were induced for osteogenic differentiation for 7 days. The treatments included active form of vitamin D3 and CSR agonist L-tryptophan, and also CSR antagonist NPS 2143. And then we measured mesenchymal stem cell mineralization and alkaline phosphatase activity, and also osteogenesis related gene expression and calcium signaling gene expression. Well, the results part showed that in broiler mesenchymal stem cells, 5 millimole L-tryptophan significantly promoted the mineralization process during a 7-day osteogenic differentiation compared with the control. But the negative allosteric modulator of CSR, MPS 2143, significantly offset this effect. Both 1 nanomole 125 vitamin D3 and 2.5 millimole calcium alone did not affect the mineralization level of osteogenic differentiated mesenchymal stem cells compared with the control. Well, in broiler mesenchymal stem cells, similar to ARS staining, the 5 millimole L-tryptophan group showed the highest alkaline phosphatase activity, and NPS 2143 did not affect the effect of 5 millimole L-tryptophan on ALP activity. In addition, 1 nanomole 125 vitamin D3 supplementation significantly inhibited ALP activity compared with the control. And in contrast, in layer mesenchymal stem cells, 7-day differentiation obviously facilitates the calcium deposition among all treatments. The highest level of mineralization was detected in the 2.5 millimole calcium alone treatment group. 5 millimole L-tryptophan did not promote calcium deposit. On the contrary, 5 millimole L-tryptophan alone and 5 millimole L-tryptophan plus NPS 2143 significantly inhibited the process. Well, in layer mesenchymal stem cells, ALP activity was also faster than that in broiler cells. Consistent with its ARS staining results, the highest staining value was found in the 2.5 millimole calcium group. 5 millimole L-tryptophan alone and 5 millimole L-tryptophan plus NPS 2143 significantly inhibited the ALP activity. Well, the mRNA expression of most genes were upregulated by 5 millimole L-tryptophan compared with the control. NPS 2143 alone only promoted the gene expression of OPN, BMP2, and RUNX2, which were significantly higher than L-tryptophan alone. And the gene expression of OPN and BMP2 were significantly higher in L-tryptophan containing groups compared with other groups. NF80C1 gene expression did not show significant difference among all treatment groups. In layer mesenchymal stem cells, regarding the gene expression of CSR, VDR, ALP collagenase 2, NF80C1, and beta-catenin, no treatment group significantly upregulated the expression level compared with the control. Well, for the gene expression of OPN, OCN, BMP2, and RUNX2, at least one treatment group showed the significantly higher expression level compared with the control. Well, to summarize, CSR is expressed in broiler and layer mesenchymal stem cells, and osteogenic differentiation in layer mesenchymal stem cells was faster than that in broiler cells. And CSR agonist by millimole L-tryptophan promoted osteogenic differentiation in broiler mesenchymal stem cells for 7 days. And the 1 nanomole 125 dihydroxycalciferol did not facilitate the differentiation and mineralization process in chicken mesenchymal stem cells. Well, now we know chicken CSR we're studying to mediate bone-related diseases. Therefore, in the future, we will focus on CSR and investigate it if the effective CSR nutritional ligands are able to improve bone health and actual quality in vivo. After that, we will explore the effect of CSR modulators, the temporal sequence of osteogenesis, and the downstream signaling pathway after CSR activation and during chicken osteogenic differentiation. Well, at last, we really appreciate the support from UNSERC, University of Manitoba, Manitoba Egg Farmers, and the Wallenstein Feed and Supply Company. Thank you for listening, and I'm happy to take all questions.
Great, thank you very much, Qianru. That was very, very interesting. I'm going to I'm going to betray my ignorance when it comes to this sort of thing. I am not a lab person. I admitted that in our technical meeting before we went live with everyone. I'm much more about the extension and about bringing people together in meetings like this. So my question for you is, when you're doing, you talked about the different cell, you're looking for a very specific type of cell when you're doing this work. How do you know you've gotten the right one?
Oh, that's a good question. Yes. For sure, because the mesenchymal stem cells, it has definition and also several criteria to identify mesenchymal stem cells. So in my study, I showed the results of cell surface markers, but I haven't shown other criteria because of the time is limited to present these results. And so the first one is the capacity to adhere to a plastic surface under standard cultural condition. So when I isolate these cells, I make sure, yes, it stick to the plastic surface. And also it need to have a multi-lineage differentiation property. So in study, I specifically tested the osteogenic differentiation ability. So it's still good. And also, like I mentioned, I tested the presence and absence of surface antigens. And also I need to mention that, the cells grown up from a single colony. So it's also the other method to make sure the purity. That's a very base for this study, because if without the correct cells, without the pure cells, I cannot conduct the rest of the study, right?
Okay.
Yeah. Thank you.
Very good. Thank you. Thank you very much for that answer. I did have a question come in, but unfortunately we're at the end of the time for your presentation and your answering. If you'd like to type an answer to the question, if you can see it in the Q&A, that would be great. If you need help with that, just send Kyla or I a chat about how you can do that. So thank you very much, Qianru. I appreciate your presentation very much, and wish you all the best as you continue your research.